Hello, I'm going to tell you a little bit about some SFARI updates for 2021. And just to remind you and give you a little background on the Simons Foundation, which is the parent group that supports the Simons Foundation Autism Research Initiative. So the Simons Foundation is a private foundation based in New York City that was founded by Jim and Marilyn Simons. It has a very large endowment and supports work across all areas of mathematics and physical sciences, life sciences. There's a new computational group called the Flatiron Institute, and there's a very active outreach and education group. And I encourage you to look for information about all of this off the Simons Foundation website. But everything else that we're going to talk about today here has to do with the Autism Research Initiative, which we call SFARI. SFARI has a budget of about $100 million in 2021. And on the following slides, we'll give you some updates on what we've been up to. One thing of particular interest to researchers are the various types of grant opportunities we have. Several of these grant opportunities are currently closed for 2021. Their deadlines have just closed, but I want to give you a brief overview because each year we have a different types of RFAs that we launch. And if you keep your eyes out on safari.org or our website, you'll get to follow those. We have a pilot award program, which is to encourage investigators who are new to this autism neurodevelopmental space. These are grants for 2 years, up a total of $300,000. We have a Bridge to Independence program, which is a really creative and important program that helps investigators working in this space who are transitioning from postdoc to becoming independent investigators. And I encourage you to follow that. That's been a yearly program and will continue as such. This year, we had a new RFA called Human Cognitive Behavioral Science. And one of the goals of that was to encourage and fund more work in humans, largely making use of some of the cohorts that I'm going to tell you about on the next slide. And then lastly, we have an RFA that's still open called Genomics of ASD Pathways to Genetic Therapies. And our goal there is to fund research in this exciting, very active field that moves from an understanding of the genetics towards potential targeted therapies, potentially involving antisense oligos or gene therapies or various versions of that theme. We spend a lot of our time, energy, and funding on building large cohorts of deeply characterized, deeply phenotyped individuals. The two that I want to point out here that have been most active this year are SPARK, which is a very large study that recruits individuals with autism. The goal is to have 50,000 families with autism. In certain ways, the project has already exceeded that goal, but it's I encourage you to learn more about this. It's open to all families in the United States with a medical diagnosis of autism. It's free to join. There's a genetic testing component of it. And what I really want to emphasize, along with all of the Simons cohorts, is that there's a really an emphasis on rigorous phenotypic and genetic characterization. Many of these cohorts are characterized both at the array CGH, the whole exome sequencing, and the whole genome sequencing level. Not SPARK, not whole genome, but I encourage you to learn more about that. And a key part about these resources that we build, these cohorts that we build, is that they're really intended as a community resource. They're open for investigators around the world to use. And the other cohort I just want to point out here is Simons Searchlight. Simons Searchlight recruits, instead of on a behavioral diagnosis of autism, on a diagnosis of an individual having a genetic change in one of a now quite long list of recurrent genetic conditions. And again, our aim is to involve families in research and to connect them to the research community writ large. Another major goal of SFARI is to lower the barriers of entry and to bring new scientists into the field. And to do that, we fund, maintain, and curate a number of resources. Those include resources of mouse, rat, and zebrafish models, to enable labs that can't generate these on their own to be able to get into this research area. From several of our cohorts, we've generated a large and now growing number of induced pluripotent stem cells, or iPS cells, and those are available to the research community. And we also have a collection of post-mortem brain tissue for those interested in studying the brain of humans. We also maintain certain tools such as SFARI Gene and SFARI Base to help investigators navigate what is a rather complex genetic, a source of genetic information. And also SFARI Base is the go-to place for all Simon's data relating to autism and neurodevelopmental disorders. And on some of the following slides, my colleagues will go into more detail about various of these tools. Thank you.
One way SFARI attempts to lower the barrier of entry to study the diverse genes linked to autism is by creating, or at least compiling, animal models and cell line models that are representative of the various genetic changes. SFARI makes those models available for request by researchers through various repositories. For mouse models, SFARI is working with Jackson Labs, and those mouse models that SFARI helped to make available are listed on this slide. Some prominent autism mouse models were available via JAX or other repositories prior to SFARI starting this effort, as for example the neuroligin 3 models, and therefore we didn't add them to SFARI's specific repository. Any researcher can order the mouse lines listed here from JAX. More information can also be found on safari.org under the resources link. A somewhat more recent effort is to establish a repository for rat models of autism. For that, SFARI partners with the Medical College of Wisconsin to create knockout rats, and with the Simons Initiative for the Developing Brain at the University of Edinburgh to do some basic phenotyping, the data of which will be made available to the research community. MCW uses CRISPR-Cas9 technology to create knockouts, and the rats are maintained on the outbred long Evans background. 8 rat strains are currently available for order and are listed here, and one more line is expected to be available soon. Again, to learn more about this growing resource and how to order rats, please visit safari.org resources. Complementary to the animal model resources, SFARI is creating human-induced pluripotent stem cell lines from the blood of participants of the Simons Searchlight study. For this effort, SFARI partners with the New York Stem Cell Foundation and the biorepository IBX. The iPS cell lines are derived from peripheral blood mononuclear cells, PBMCs, with Sendai virus delivery of the reprogramming factors, and the iPS cells undergo a rigorous QC process prior to release and after every round of expansion. We currently have iPS cells from individuals with genetic variants in 2 autism-linked genes and 1 autism-linked CNV available. Upcoming are iPS cell lines from individuals who carry variants in 1 of 13 different autism-linked genes and 1 additional CNV. For each of the genes or CNVs, lines from multiple individuals that carry different variants will be available. The resource will continue to grow, and we are planning on adding more iPS cell lines for additional genes. We are currently not making isogenic control lines or family member control lines, but we are considering doing so, in particular if they are necessary for SFARI-funded projects that make use of the patient lines. And here I should also mention that the selection of genes for which to create iPS cells, although contingent on the availability of blood samples, is flexible to some degree, and one determining factor is researcher interest, as we want to make sure that we're creating resource that is needed and used. So if your gene of interest is not listed on this table yet, but you are interested in iPS cells, I'd encourage you to reach out to us at iPSC@safari.org, and we can look into the possibility of creating iPS cells for your gene of interest. Again, you can find more detailed information on iPS cell models, as well as on how to order them, on the safari.org website under the resources link.
Hey, I'm going to give you a quick demo of SFARI Base today. So as you can see in my account, I'm a newly created PI. I don't have any requests and I have one existing project, but I'll start with up here in the corner and placing a new request. So since I already have an existing project, I can either choose to pre-populate it or I can create a new project. Today, I'll just create a new project starting from scratch. The first details they ask you for scientific title and abstract. So I'm going to copy and paste from another Word document. I just have this sample title and abstract in. The next question, it asks if the project is related to the field of autism and related neurodevelopmental disorder research, since certain cohorts have limits due to their consents and what the data can be used for. So just answer appropriately. We also ask for a general audience title and abstract in case we are posting it online. So something that might be more appropriate to share publicly, as well as considering communication, like if it's written in plain language instead. So you can either use your scientific title displayed above, or you can use, you can provide a different one that you would be comfortable with sharing publicly. So I'm going to say that it's okay to use the one above. Here is the research team section. You're able to add any other colleagues or collaborators that will need the data. Please note that if you are adding individuals who are other co-investors or collaborator levels, they will also be asked to make a SFARI Base account if it doesn't already exist. And they will be asked to provide their own IRB due to compliance reasons. However, if they're individuals in your lab, such as lab managers or postdocs, students in your lab, they'll be covered under your IRB. So feel free to add them here as well. So here we get to the resources sections. You can either view by resource type, or if you're interested in a specific cohort, you can also filter by that. So filtering by cohort, I'll look at Simon's Searchlight and I'll see what's available. So these are all the data sets available. It includes phenotype data, biospecimens, as well as genetic data. So I will choose these two data sets as pheno data sets that I'm interested in. If you have any questions about the data sets, you can always click more info. There are also links to researcher welcome packets with more information here. And as always, if you have any questions about any of the data sets or any issues with the process, you can contact us at collections@safari.org. So the next step, continuing on, I selected my resources. Now it's asking for IRB exemption or approval letter, and these are required in order to access our resources, again, due to compliance reasons. So I will put, I have an exempt project. I'm going to choose my IRB example doc here. And I will put in, you have your approval number, but I'm not sure what it is since it's just an example. So I'll just put in that approval number. And then my exempt project, I'll say it was approved in February. We also ask you to abide by NIH security best practices and just agree to storing the data securely. So feel free to review. This links out to the document with best practices. So here we also, if you can once again have another chance to add team members if necessary. So as I mentioned before, you can add anyone else who will need access to the data, but collaborators and co-investigators will need to provide their own IRBs and complete this and also register on SFARI Base. Before we proceed, we ask you to complete a little quiz just to make sure you understand limitations to sharing data as well as limitations to data use. So I'm just going to breeze through this quickly, but we do ask that you read that and agree to the terms. The last page just lets you review a summary of what project you're using, which resources you are requesting, as well as the research team members. So after that, we get a confirmation page. You should also get a confirmation email. If you don't, please contact us so we can look into things and see what's happened. Then you're able to view your request details. For phenotype data sets, after you're approved, the information will come up under access details. So right now it's blank here. For genetic data sets, we do ask that you provide us with Globus IDs. So we're able to provide access after you're approved. So if we go back to our homepage, we can see that this is just submitted, and when it's approved, the status will change. And throughout the whole time, you'll receive email confirmations from our team as well to let you know what the status is. If we have any questions about your abstract, if the reviewers would like more detail on something, we also reach out. So keep your eye out from any emails from collections@safari.org.
My name is Casey White. I'm a project manager at SFARI, and I'm here today to talk to you all a little bit about Research Match. So Research Match is a service that matches our SFARI cohort participants with approved research studies. These are studies that are external to the cohort themselves, maybe from university professors who have a project that they'd like to do. They can apply and receive access to recruit our participants into their new study. These can be in clinic studies, online survey studies. Sometimes we'll build these online survey studies for the researcher, or sometimes we'll hand off to the researcher so they can complete your own workflow. We also do remote or virtual participation, things like focus groups. We can help you recruit for those, or remote sort of studies where you have them doing some sort of a task on an iPad or something similar from a remote location, or collecting samples. From its launch in July 2017 until April 2021, for Spark, one of our other SFARI cohorts, we've approved over 150 research studies. And of these, we've launched about 80 and have more than 70 of them in progress. So this is a process that we're very familiar with. We've been doing quite a bit of for one of our other cohorts. We've invited more than 80,000 of our Spark families into new research studies. And a lot of these families have been invited or have even participated more than once. We even have families who have done 10 or more studies for Spark researchers who were approved and did a research match study with us. More than 33,000 families have responded to at least one invitation. And researchers have published 12 articles and counting from studies that are supported by Research Match. And so over the cycle of the study, we recruit and they gather data, then they analyze and publish the data. And eventually we sort of see this coming full circle in terms of accelerating research and seeing new articles published. So Research Match for Simon's Searchlight was actually just launched in December of 2020. We've already completed the first project from start to finish. And so as of May 2021, we have 3 additional researchers who have applied and been approved, and we're currently preparing those for launch. We're expecting one of those to launch within the next month and probably 2 more over the summer. And I'm anticipating we're going to continue to see these applications on a regular basis in order to use the service and recruit Searchlight participants. And we hope we'll continue to accelerate research even above and beyond what we're doing here at SFARI. Researchers who are interested in accessing Research Match may apply on SFARI Base. So SFARI Base is sort of the clearinghouse for all of the resources related to the SFARI cohort. So if you've accessed Searchlight data or biospecimens, you're probably already registered on SFARI Base. If you've never done that before, then all you have to do is to go in and create an account, affiliate with your institution, and sign a joinder to the researcher distribution agreement, which is signed by your institution. And then you can place an application for any of the resources, including Research Match. The application does require you to have an approved IRB protocol and consents in place. We can make some modifications later as we discuss the Research Match process, but just have something kind of ready to go for us to review. The application will also ask you for some additional information, including your study aims, inclusion and exclusion criteria for your study, whether or not you're providing an incentive or how much time it's going to take participants to complete, any feedback reports you may give, in addition to some other questions. Currently for Searchlight, we're approving applications on a rolling basis. So when you submit, we review it and summarize and may ask you a few questions, and then we refer it on to a scientific committee pretty quickly. So usually you'll see approval within about 3 to 4 weeks of when you submit your application. The committee who reviews the application considers criteria such as the scientific merit of the proposed study, alignment with the Simon Searchlight mission, burden and engagement of participants as they complete the study, sort of what's being asked of them and what's being given back. Once a study is approved, the research match team will schedule a conference call with you to begin to discuss your project details and next steps. And at that point, you'll get lots more information about your specific study, and we'll work with you to draft any materials that are needed for us to build your workflow and get everything launched. Usually we can launch within about 2 to 6 months of approval. It varies by the complexity of the study and also how long your IRB takes to approve any modifications, as well as the research match team sort of load at the time. But typically within 2 to 6 months for a study team that's sort of doing constant work on the study, we're able to get those launched for you. And so I hope this gave you sort of a general overview of research match. If you have any questions at all about research match, you can reach our experts at collections@sfari.org, and we'll be happy to help you out.